Okay, thank you very much, Miguel. Awesome talk, by the way. That was really cool. And so what I'm going to talk about is a few other things that are the result of my attempt to find some new things that are not just old results. In other words, we haven't been doing anything really with entangled states and things like that. We've been using simple quantum systems for a long time. And on the other hand, I think there are a few things that I might be able to just mention to this audience that might be interesting. So the idea here is, here's the overview. I'm going to give a little bit of early history. Some people may not have seen some of the early single molecule imaging spectra, vibrational modes, and the ODMR that year, those years. The surprise in 1997, which is this blinking process, and EYFP. And EYFP keeps coming back a little bit because that's also this protein that was used for this ODMR advance by Fader et al. That was reported at the beginning of the meeting. And I'm going to talk also about some super resolution imaging with single molecules, specifically or primarily SARS-CoV-2. Because I think it's an amazing demonstration of what you can do with those simple methods. Then I'll talk about combining single molecules with electron microscopy and correlative imaging. Oh, Pete, are you here? No, the other Pete. Peter is this one here, but this is Peter Dahlberg, who was going to try to make it, but since he works at SLAC now, there's a whole bunch of DOE meetings, and so he couldn't make it. Anyway, yeah, I'll tell you about what he did and how that relates to what might be interesting. End up with a special fluorescent protein at 80K that one of my students looked at, which might also be a potential candidate for these fluorescent protein ODMRs. Okay, so way back to very early days, 1991, all right? So 1991, 2K, here is the inhomogeneously broadened absorption line of pinosine and paraterphenyl. This transition is measured by basically what we call fluorescence excitation. You tune the pumping laser and you collect and see how much fluorescence comes out of the sample. So that's what this is. And the center here is at 506 terahertz. So you look at all of these features and you zoom in here on just a couple of gigahertz, even here over 100 megahertz. You see these unbelievable spikes, which are the individual single molecules coming into resonance with this single frequency laser as you tune over them. So this, of course, it sort of started out this field. Here's some imaging with single molecules back in that same year. Frequency on one axis, because frequency is a good variable, and space on the other axis. And so the peaks that you're looking at here in the space axis are the molecules measuring the shape of the laser spot. So now what else happened is that other people started doing this imaging. Here's Urs Wild doing that in 1994. But there's a whole story of those first years at low temperatures. I just want to emphasize one thing. Imagine that you tune in to one of these single molecules. You just set your laser at that frequency. And if you do that, then you can then disperse the emitted fluorescence. And this is an example of doing the vibrational spectroscopy on that single molecule. We'll change the molecule, but that doesn't matter. You're into resonance with one molecule and you disperse the emitted light and you see these spectra, which of course are the even vibrational modes of the electronic ground state. So these are different in different local environments for the different single molecules. Here's one at one laser pumping frequency, and here's one at another pumping laser frequency. So that's another whole story. But you can also sit on that molecule's emission and do ODMR. So this is what was talked about earlier. This was in 1993, done in my lab with Jürgen Köhler, at the same time that Jörg Wachtrup and Michel Ary were doing it in Europe. And so it was really a race. These papers are back-to-back in Nature. And the idea is well known to this audience. We're using the fact that intersystem crossing will populate the triplet state. Normally, that's a bottleneck that takes the emitting molecules into dark. But those that triplet state has triplet spin sublevels that populate differently because of the symmetry of the molecule, actually, and the electronic structure. And so there's a high probability of being here and then less, but fast rates, slower rates, leaving those states. So all of that put together will give you an average triplet lifetime. Once you're in the triplet state, depending upon what you picked up on those numbers, what will be the triplet lifetime. So the triplet lifetime will change in principle as you go into the triplet state. And that's why these ODMR spectra, where you're tuning the microwave frequency, okay, over this range here from 1475 to 1500 megahertz, has this tail on it. So that's the idea for sensing these magnetic resonances. And after this experiment, Jörg Köhler and also Jürgen Köhler and Jörg Wachtrup, really, I'm very sorry to screw up those pronunciations, went to town on all kinds of amazing things: spin coherence, Zeeman effects, isotope effects, indoor, and all kinds of stuff. So many demonstrations with this single molecular spin. So very nice. So okay, so now let's go a little farther forward to room temperature. So all of that was 2K. Now we're at room temperature. We wanted to see what happened. Rob Dixon wanted to see what happened for a single fluorescent protein. Can we detect it? No one had detected it optically before this time. And so we got some material from Roger Chin's postdoc at UC San Diego and put it in a gel. And so this particular fluorescent protein is EYFP, so what basically Rob observed in those gels is that you could indeed see single fluorescent proteins at room temperature, but immediately blinking appeared. That is, the molecules would be on for successive 100 millisecond frames, emitting light, and then turn off for a long time, seconds or two, but then coming back on, spontaneously, and then off and then on. And so this is basically some process where the molecule is cycling, giving you light, and then goes into an intermediate state, but can thermally come back from that intermediate state and start emitting again. So this is a reaction of the chromophore I'll mention in a moment. But after watching this for a while, it completely stops. After a long time, completely stops. Maybe that's another barrier going into a deeper dark state. And one thing that Rob learned is that if you could turn on 405 nanometer light, shorter wavelengths, and restore the molecule from that dark state and have it emitting again, and then do it again and do it again. So we call this basically photo recovery of molecules that have been put into a very long dark state. So now you got to keep in your mind a bunch of time scales, okay? So this is not confusing. There's a really, really long time scale, and then there's this blinking time scale of milliseconds to seconds, hundreds of milliseconds to seconds. And then, of course, there's the triplet lifetime, which we heard about earlier, and we'll talk about more in a minute or two. Anyhow, there's been a great deal of work on many different switchable fluorescent proteins by a huge number of people, groups all over the world. To try to change wavelengths, have different behaviors, have situations where it's very reversible, like DRAMPA. But it's likely to be an isomerization of the GFP chromophore, perhaps, and also maybe some deprotonation. And so this is quite a wonderful thing, okay, that you have not seen probably. From that time, this picture came out in Photonic Spectra, okay, which is one of those wonderful sort of journals back in that time, right? Optics journals, trade journal. And the article is sort of saying, what's with these crazy scientists? They're going to be storing data in jellyfish. And so anybody thinking about fluorescent proteins, this is a funny one from long ago. All right, so back to the main story. This blinking that I just showed you at room temperature for EYFP, I just want to show it to you a slightly different way. This is now in cells. So here's a bunch of bacterial cells where EYFP has been fused to a specific bacterial protein. And the wonderful part about it is just to watch this movie of the fluorescence. So nothing's moving here. Nothing's moving. These molecules are not moving around in the cells on this time scale. All of this is the blinking of these fluorescent proteins inside those bacteria. And what you notice is that it's pretty fast here. The movie is slowed down 3x. Turns out that the blinking will be faster if you use different laser powers and things like that. So there's some variation in detail about the blinking processes. Fine. no time to go over that. Here's an example of using EYFP blinking, 3 examples of using EYFP blinking to do super resolution. So now here, the idea is, as everybody knows, we're going to use super resolution in the blinking to localize different molecules at different times and then show all of their positions all at once, right? To go beyond the diffraction limit by the old methods. The new method sounds so exciting. But in any case, this is MREB, this is PAR A, and this is HU. And you can see that the diffraction limited images don't tell you much, but these super resolution images show a lot. That's the reason that this is, of course, interesting. To be able to go beyond the diffraction limit by factors of 4 or 5 is not a small effect, okay? Fine, now I'll show you a more modern example of super resolution, just because it's a really very recent work, and I think it's very exciting because we also were affected by the pandemic and so on, and so we wanted to do something important, as important as possible during the pandemic. So let's look at SARS-CoV-2, the SARS-CoV-2 virus as it infects cells. So this timer doesn't work, so sorry about that. Hopefully I'm not running over too much. 17 minutes left? Yes. Good. Okay, so the SARS-CoV-2 virus, the virion, the structure of the virus particle, is very small, 120 nanometers or so, and it has this spike protein all over the surface that you've heard about that binds to our cells. But inside it has the RNA for the virus. It's basically an RNA virus, because this is a single-stranded piece of RNA that's been decorated by these nucleocapsid proteins, these N proteins. Okay, so that's all it takes to infect someone. And so the viral genome is what we were really focusing in on here, because the neat thing about this genome is that it's a positive sense single-stranded RNA. That means it's ribosome ready. As soon as it gets into the host cell, the ribosomes can start making proteins. And then it's all those proteins that are the rest of the infection. So we want to look at viral genomic RNA. We want to look at certain numbers of the proteins. So this is the gene itself, and there's different what are called non-structural proteins and structural proteins. Here's spike. Spike has to be made, etc. Okay, so there's in fact 29 proteins total. That's all, though. That's all, only 29 proteins. And I'm really now talking about the work of Leonid Andronov, a postdoc in my lab, in collaboration with Mingting Han and Stanley Chi's lab. So Stanley Chi is in the bioengineering here at Stanford and is our biological partner on all this work. Okay, so and only a small amount. I'm going to show you only a small amount, and maybe encourage you to take a look at the papers. First, let's just look at the virions themselves. Okay, the virions. The virus particles. And now we're going to label, of course. We can label the viral genomic RNA with FISH, fluorescence in situ hybridization, given with a blinking dye, Alexafluor 647. So one of these dyes that blinks, it gives you super resolution, okay? So now I'm switching away from fluorescent proteins to dyes, which actually are better blinkers than EYFP, but anyway, that's what you want to do, go to the better ones. Spike is labeled also with a blinking dye, CF568, but it's basically an antibody. So this is immunofluorescence to light up certain things. All right, so those two together show these structures in the diffraction limit. But if you now go to super resolution and look at just one of them, you begin to see interesting structure. And here's multiple copies of that. Notice this is 1 micron scale bar. This is a 100 nanometer scale bar. So the super resolution shows a lot more detail. And you see these concentric structures. The inner part is the magenta. That's the viral genomic RNA inside that virion, like I showed. And the spike is around the outside. So that's just showing we're basically able to do this in a reasonable way with a certain amount of resolution, down to, well, let's say 20 to 40 or so nanometers. Okay, now in the cell, originally we had to work in BSL-3 conditions and get the samples taken care of there and then fixed and brought out of BSL-3 so we could image. So we're really working on fixed cells, which are much less dangerous, of course, than living cells. And now I'm showing in magenta the nucleocapsid protein, that N protein that wraps around the viral genomic RNA, as well as spike in green, near the edge of the cell. So the edge of the cell, you see, is decorated with a huge number of filopodia and lots of viral particles, because you see many of these little spots you see have green as well as magenta. So this is an idea that by using this imaging, we start to see what's going on in the cell. But the real interest is more near the nucleus for this first set of experiments, because now looking at just the VgRNA in one color, and the coloring is, of course, the something roughly corresponding to how many blinks were there. These are the nuclei. This is a big scale. And what do you see when you look at the RNA? You see little clusters all around the nucleus, perinuclear clusters. So in diffraction limited, you don't see that at all. You just see blurs. And if you look in at specific ones, here's a region that here's a few clusters here, different examples of clusters. And down here is an example away from all of these clusters, just showing very small puncta, which are probably loose single RNAs from the genome. So my point here is that a tremendous amount can be learned just by this simple super resolution imaging. Now, and let me just remind everyone that all of that data, those beautiful images, are coming right from this kind of raw data. This is the raw data looking at one of the two fluors, in this case VgRNA. It's that amazing blinking that can give rise to these beautiful images. And so let's also note that there's many other proteins to study. So we have a new paper. There's a massive atlas of all protein and RNA targets in infected cells. It's now on bioRxiv. So Leonid Andronov led a second project where we went in and he looked for all of the proteins and where they are. Interesting localization patterns throughout. Okay, so anyway, that's one of these applications. Well, now you heard. Let's go back to the beginning of this meeting. Okay, when Peter very nicely described his new work and Fader's work and all the other collaborators on EYFP as a molecular spin qubit, and its ODMR, basically spectra, come from, again, taking advantage of the triplet spin sublevels. And I'll mention some of the special features in a moment, but the ODMR itself is shown in this plot and in these plots. So this is basically the microwave frequency, the RF frequency, as it's being scanned on this axis, and on this axis is the applied field. You can work in zero field if you want to. You don't need to apply a field, but if you do apply a field, you see the shifting of the resonances and so on. So at a specific single field, you see these dips in the emission, just like I showed you in the pinosine paratorphenyl case. What's going on is that the RF radiation, when it's in resonance, changes that mixture of over the 3 spin sublevels, so that when you do that, you alter the triplet lifetime, and therefore you alter how much light can be detected back in the fluorescence channel. So so it's a beautiful experiment, and I was so excited to see that they had done this since EYFP had been used for the other work earlier. And so beautiful experiment. There's also in their paper a discussion of why they use this 912 nanometer readout or assistance to the readout, because things were too slow. They want to go faster. You're basically motivated to get information about spin substates, et cetera, on a much higher rate than if you didn't use this extra light source. And what it's doing is pumping T1, Tn, or T1, T2 absorptions, so that the molecule can, by reverse intersystem crossing, get back to the singlet states faster than it would if it was left to sit in a triplet state. Okay, so that's great. That's wonderful. This is something also called OADF by Rob Dixon, optically activated delayed fluorescence. But essentially, it's beginning to use the different colors of light that you might have available to do more gymnastics with this emission. And so that's why they used the 912 nanometer light to read out on a faster time scale what the situation was with the spins, essentially. And so all kinds of neat things were done, including doing it inside cells. So I think this has a lot of promise. But in the context of what I was saying, remember that these fluorescent proteins not only do things on these sorts of time scales, and in particular the time scale that is important for the triplet state itself was about 5 milliseconds for that EYFP that they reported. But if you, if you remember that I said that these YFPs on a longer time scale would just completely turn off because of change in the chromophore completely. And so that will stop experiments, of course, and even for a longer time scale, it needs to be restored back. So there's a great need for getting more control, if you like, over the electronic states of these interesting emitters. And at the same time, there's all this beautiful manipulation of the spins. So now I want to do another small aside because it's going to be important in what I'm going to say. In the end, I want to ask you a question. Here's one of these beautiful cryo-EM images of a cell. This is a bacterial cell. Here's a scale bar, 200 nanometers. And so you see a block. What do you see there? Well, you see some proteins, but the question is which proteins are which? Which proteins are what in that complicated image? You're going to recognize ribosomes, maybe a polar condensate. You can recognize various layers that are in the surface of this particular bacterial cell, and so on. But people will then annotate these kind of images by hand or use programs, but where are the other thousands of hidden proteins that you can't recognize with your eye? Where are they? So that's the question to be solved. We want to do that with fluorescence. So this is the workflow that Peter Dahlberg put together for correlating single molecule fluorescence localizations and cryo-ET images. Basically, what you do is you fluorescently label the protein of interest and then plunge freeze it as usual for on a grid for electron microscopy. But then, keeping it all in liquid nitrogen, we took it to our low temperature microscope so we could do imaging of the cells themselves and pumping and collecting emission, et cetera, from the fluorescent labels. And so then there's also some fiducials in these samples. Then you take it to the $5 million electron microscope and image at low mag and then at high mag and use fiducials to connect the two together to do image registration and visualization. So that's all great, that's fine, but notice there's something kind of important here. If you're going to do this with a fluorescent protein label attached to the protein of interest, it has to be able to do well as an emitter, okay, at 77 Kelvin or 80 Kelvin, because all of these measurements are done at that low temperature. It's not room temperature anymore. So a lot of things change. And so what we found in the early work is that a particular fluorescent protein called PAM-Kate, here's its chromophore. PA means it's photoactivatable. So it's dark completely until you give it some 405 nanometer light, which converts to the emissive fluorophore. That's this anionic form of the chromophore. And notice that there was not a lot of conformational change when you did that, okay? There's other fluorescent proteins that do have a big conformational change, but they didn't work at low temperatures because you have frozen ice and everything not moving around this chromophore, and so it cannot undergo its change to produce the other form. But PAM-Kate is good in that respect. It's also a red fluorescent protein. And we like that too because that means there's less autofluorescence from the samples. Okay, so Pete, then here's I'm showing you also the absorption and emission bands for PAM-Kate and the absorption before photoactivation, before this step. So now let's try to use this because the photoactivation also allows you to just turn on a few fluorescent proteins at low temperatures, okay? And not have them all on at the beginning. And so that's what Pete did to demonstrate this correlative effect. Let me just set you up for a second. Here's here's the bacterium, and the label PAMK is attached to MCPA, which are the proteins that form this chemosensor, which is close to the membrane. And this was really a control because you can see it in the EM image. You see these lines here. That's a slice through the chemoreceptor array. This is what you get from diffraction-limited imaging. Of course, you can't localize anything very well. And here's that receptor array there. But when you switch to super resolution, when you switch to localizing the single molecules well, then you get much more information about the locations of the proteins. And this is an example of the correlative work that can be done. The chemoreceptor array was a proof that the light is coming from where it's supposed to. Pete also did one more important experiment, and it turns out that the light that you were using to image the single molecules was causing basically some devitrification of the glass, of the ice glass that the sample was trapped in. And so by going to advanced metallic grids, then you can get many more localizations in much less time because now you can use a little bit higher laser power without causing devitrification. So anyway, there's a real correlative thing available here. And so now let's go back to the connection to EYFP and Peter's work. What about PAMK? Can it be used for spin experiments? And so Davis Perez spent some time up until 2024 when he graduated, taking a look at PAMK. And we wanted to control it. How bad am I doing?
You got a couple minutes.
So he started doing some transient experiments on the fluorophores itself and found that there's recovery of the emission on a time scale of about 2.1, 2.0, 2.3 milliseconds. So it's quite likely that this system has the similar type of triplet state as an EYFP. And in fact, he explored this complex energy level diagram. I'll walk you through parts of it. So this is the pumping wavelength to detect fluorescence from PAMK after it's been generated. And so you collect that emission when you put on this shorter wavelength, 594 nanometers. It's also known that you could use 405 to restore molecules from the very long-lived dark state over here through this gray line, getting back over to the emissive state. So you have a way to turn things on, but what you did not have was a way to turn them off. Many of these fluorescent proteins would just sit there forever and just emit, and then at this low temperature, you want to move to the next case, you want to try to do better super resolution, so you move from one to the next. So we found in this experiment that if you turn them both on at the same time, this yellow light as well as this violet light, you actually greatly shorten the time in the dark states and get more and more blinking and information. So what he did is to do the same experiment now, but with greatly improved emission and control using this extra wavelength. And the combination of the two wavelengths helped tremendously. 7 minutes of imaging for this compared to 3 hours in the old way. So finally, imagine, since these are single molecules of PAMK in this bacterial system, that we can do some manipulation to control how long they last. What if you did single spin magnetic resonance on that molecule? I think it would be fascinating to be able to use and measure B at each location inside the cell, the magnetic field. And, I think this might be possible, although, it has some challenges associated with it. But anyway, that's my thought about it. I want to thank my people, my collaborators, my group, etc. And you might notice that you see my group has had something to do with these big stone balls that are out in the courtyard here. Okay, thank you.
Thank you.
Is it? Oh, okay. It's working now. Sorry. So just a quick question. I'm wondering, so how many frames of image do you need to get for that blinking super resolution image? Like how many frames do you need to get to start processing?
Well, it depends upon how well you want to know the localization of the molecule. So because we combine those photons from different images to get better and better localization precision. So, you know, in these cases, it's probably 20 or 30 frames, something like that. The point is that, however, at this low temperature, you do get plenty of photons if you haven't turned the molecule off, and you can get 9 nanometer or better localization precision in these cells for these kind of measurements.
Thank you.
Hey, Remy.
Hello, boss.
Yes.
I have a question about the devitrification story.
Yeah.
So did you guys, I have not read that paper, I'm sorry.
Why not?
The mechanism of devitrification, is it thermalization of the electronic excited states that ultimately causes that? Or is it some kind of direct interaction between the visible photons in the medium?
Yeah, it's direct heating. It's really what it is.
Okay.
So the first grids were carbon grids, which obviously absorb light.
Oh,
so that was particularly bad.
Okay.
Okay. So, but if you go to metallic grids, they absorb light much less.
Got it.
So you can the fact that you have that high thermal conductivity and so forth there available, that causes the extra light to get propagated away fast enough before it can cause devitrification.
Got it. Thank you.
Yeah.
We'll take one more question.
Yeah, quick question, Louis. Thank you.
Hey.
My question is about the PAM Kate.
Yeah.
You did say that the chrome 4 doesn't really change the structure.
Right.
But I noticed there's a decarboxylation, if I got it right. Is that some kind of general theme that you can actually make all the fluorescence proteins, or at least red type of fluorescence proteins, to become photoactivatable at cryogenic temperature?
Okay, so yeah, it's a good question. There's a whole field of work where people are doing these experiments to generate new and different fluorescent proteins. I actually don't know exactly how they did it in this case, or what logic they used to create it. So, but on the other hand, what I was trying to show here in this picture, when I finally get to it, right here, this is the dark form of PAMK that before photoactivation. And you're right, here's this loss of a proton and CO2. Generally, these things can move away, especially if you're at room temperature, but may not move too far if you're at low temperature. But nevertheless, the protons certainly move. Everything that I was talking about is sort of downstream from this. This is only done once at the beginning, and then you are working in the emissive states. And you have the triplet, and you have the dark state that's appeared, and so on and so on. So what is the dark state? It's not absolutely clear, but if it's a reprotonation, perhaps then it's the more 405 nanometer light that's driving it back into the emissive form. So I'm saying that those two different wavelengths have multiple effects. The reading laser not only produces fluorescence, but also produces some transitions amongst all those energy levels. And the 405 is used to bring molecules back from dark. But when you have both on, then you certainly get turn off with 405 and 594.
Right.
So because that was the point of using the two colors, you could turn it off much better. Very complicated.
Yeah, sounds really complicated. I was just wondering if you can make a really general prediction of making lots and lots of fluorescence proteins into this form that would really help the field, right? Yeah.
Well, there are groups of people that do it all the time, and they may know a better answer to that.
Okay, let's take W one more time.